think that I've really gone over how immunity works. I went through the lymphatic system, but here's going to be a quick overview over the entire immune system, both the innate and adaptive body defenses. So innate are those that do not react in particular to any antigen. An antigen doesn't trigger their action. They are already there. They are already doing their job regardless of what is trying to get into the body, whereas adaptive defenses are not going to do anything until they've been stimulated by an antigen to mount an immune response. Often we refer to them as non-specific and specific defense, or first and second line of defense, and then third line of defense. So we'll take a look at all of those. So here, just objectives. We'll look at the surface membrane barriers, protective functions, phagocytosis and natural killer cells as part of the second line of defense, as well as inflammation and antimicrobial substances such as interferon and complement, how fever as a second line of defense works, and then the rest of them will get into specific defense. What is an antigen and how do they work? B and T cells, where do they come from, where do they go, and what do they do? Macrophages and the other phagocytes, what immunocompetence and self-tolerance are, humoral immunity, plasma cells and memory cells as an aspect of humoral immunity, and then active and passive humoral immunity. Then cell-mediated immunity, we will talk about antibodies. We probably won't talk about monoclonal antibodies though. And then cell-mediated immunity, and how do we activate and clonally select for T cells, which we also do for B cells, and then the functions in the body. So with the non-specific defense, like it says, it doesn't react to or target specific defenses, just specific antigens, just any antigens in general. Anything that is not foreign, anything that is foreign, that is not part of our own bodies, and essentially it's just going to keep everything at bay. So the first thing that we have, our first line of defense are our skin and mucous membranes. So physically, we have multiple layers of skin. We have keratin as a part, if we're looking at the keratinized stratified squamous epithelium that we find in our skin, and then the mucous membranes on the inside are also stratified squamous epithelium but are not keratinized. So what does keratin do? It helps provide a physical barrier to bacteria, weak acids and bases, bacterial toxins and enzymes by making our skin waterproof. So up in that stratum corneum, it's in the stratum corneum, but excuse me, in that stratum granulosum layer is where the keratohyaline granules begin, and then they're secreted up into the stratum corneum, and then prevents the entry of any substances. The skin is also acidic, and what that does is inhibit some bacterial growth or fungal growth. In particular, our natural body flora that grows on our skin does help prevent fungi from getting a foothold, because when it gets a foothold, those little rhizomes stretch out and go in between the different layers of stratum corneum, and it is very, very difficult, if you've ever had a skin fungal infection, to try to get rid of. It's nasty. Vaginal secretions also acidic, so that's one of the chemical barriers that we have in our first line of defense, is the acidity that's associated with the skin and vaginal secretions. And then sebum has chemicals toxic to bacteria and fungi as well. Stomach, hydrochloric acid is a great chemical defense against infection as a first line. As soon as you eat something, it gets down into the stomach, starts denaturing. And then the protein, the pepsin that is in our stomach, is also proteolytic, and so it's going to start digesting any protein that an antigen or some pathogen that may make us sick would bear. Our external body secretions, saliva, tears, are going to contain an enzyme called lysozyme, which targets the peptidoglycan layer of gram-positive bacteria. So if you're familiar with microbiology, on the outside of gram-positive bacteria, they have this thick layer called peptidoglycan, and lysosome specifically attacks that. Not so much with gram-negative bacteria. Gram-negative bacteria also tend to not live on the outside of us, and they have a thin peptidoglycan layer that's sandwiched between two cell walls, and so lysozyme isn't as effective against them. Mucus secreted by the mucous membranes is going to trap organisms, and then some of our mucous membranes have cilia associated with their cells, like the ones in the trachea, the pseudostratified ciliated columnar epithelium. And so that's going to help sweep the trapped material up the trachea, up into the larynx and into the pharynx, so then we can swallow it down into our stomach. And one that I'm not really going to get into is the genetic lack of receptors or inadequate supply of nutrients. The genetic lack of receptors is really an interesting thing. I'll just talk to you about that really quick. Some babies born to HIV positive women, okay, let me just put this way. If you are a baby and you have an HIV positive mother, you will be infected with HIV. Now, there's a difference between being infected with HIV and having AIDS. So some babies are infected with HIV, and we know that they're infected with HIV because they have antibodies that are present against them. However, they do not come down with AIDS because they do not have the receptor for the AIDS virus, the HIV, that is found on the CD4 or the T helper cells, called the CCR5 gene or CCR5 receptor. And so these babies have just not inherited this gene, and therefore they have a genetic lack of CCR5, and now HIV cannot infect them. And so that's one example there. And then inadequate supply of nutrients has to do with why individuals that are heterozygous for heterozygous for sickle cell anemia. So they have, the dominant trait that makes them normal, and then they have the recessive trait, which would cause some of their cells to sickle. They do not get malaria. So they are immune to malaria. And so there's some stuff about that, but we're not going to get into that. So anyway, if you want to read more about that, why do heterozygous individuals, those who possess the sickle cell trait, don't get malaria? All right, so that's our first line of defense. Our second line of defense then has 5 aspects to it. One of those is 2 groups of cells. We have the phagocytes and we have the natural killer cells. So the phagocytes come in 2 groups. We have the monocytes that will turn into macrophages once they exit the capillary, or actually the post-capillary venule is where diapedesis will happen. So they'll leave the bloodstream, and then they'll go out into the tissues, and then they turn into macrophages there, and then neutrophils. And neutrophils are also phagocytic, and they are actually the first ones that come to the site of infection. So they'll get the signal, they'll come out in massive numbers, huge numbers, and we'll get more into this a little bit later, but they'll come out and they'll flood your tissues, but they can only engulf a few antigens before they go through apoptosis. And if you're not familiar with apoptosis, that's programmed cell death. So instead of dying and becoming necrotic, they die on their own terms. And so they'll eat the germs, and then they will go through apoptosis, and that way you don't have necrosis happening. It is cell death, but it's programmed cell death, and your body has a good way of getting rid of that without toxins spilling out and that kind of thing. Macrophages, on the other hand, will come a little bit later, a couple days later, and they will live for a long time, and they will eat and eat and eat and eat. And in fact, as the neutrophils are dying and the pathogens are dying and tissue cells are dying and pus is created, the macrophages will come and eat up the pus and get rid of all of that. So they're the good ones. We also have eosinophils that will be in parasitic worm infections, some allergy stuff, and then mast cells, of course, are the mediators of allergies, which we'll get into at the end of this lecture. Okay, now in order for a phagocyte to engulf its prey, so to speak, it must adhere to the invader. And some invaders have mechanisms, they have slime layers, they have coats on the outside, capsules on the outside that make it so that the phagocyte cannot adhere. However, we have two mechanisms in our body. We have something called complement, which we'll talk about later, and antibodies. We'll also discuss their functions that will allow a process called opsonization to take place. And what opsonization is, actually means to make tasty. And what opsonization does is provide handholds for the macrophages to bind to. So once the complement proteins or the antibodies bind, then the macrophages can come along and bind to them and then eat up whatever they're eating. All right, then we have a natural killer cell, which is a type of a non-specific lymphocyte. And their job is to go around, they're part of immunosurveillance, and they're going to be similar in function to the cytotoxic T cells, but yet they are not specific. They are not antigen specific. So their actions are exactly the same as the cytotoxic T cells or the CD8 cells. They will cause lysis of virus-infected body cells and cancer cells by releasing chemicals called perforins and granzymes. And what those chemicals do is trigger the body cells. Because remember, natural killer cells and cytotoxic T cells are going to find those cells that have the MHC1 protein on the outside. And so those are going to be our body cells. And so natural killer cells and cytotoxic T cells are looking for MHC1 proteins as well as antigens. And so they will be looking at our body cells to kill that have become cancerous and virus-infected cells. Now, natural killer cells are a little bit different than cytotoxic T cells in this function. They're not looking for the MHC1, they're looking for the absence of the MHC1. And so when they find our body cells that do not have those markers on the outside that they're supposed to, then they'll trigger apoptosis in those cells. And so that's how the natural killer cells work. All right, a very important aspect of our nonspecific defense is inflammation. So this flow chart shows everything that happens in inflammation. I'm going to go through this step by step and then just look at the individual slides that I have. So what's going to happen? Tissue injury. And I don't care the nature of the tissue injury, whether you have a chance of becoming infected or not. It could be a sprained ankle. It could be a torn meniscus. Whatever. It's just something where a tissue injury has happened. And so there's two things. First, we're going to go through leukocytosis and leukocyte extravasation, and then we'll go into inflammation. So a lot of times your body doesn't know just how bad the injury is, and so it's like, well, we better get some white blood cells there to the site so that they can help out. So here's what's going to happen. Upon tissue injury, we're going to start the tissue, or the injured tissue cells are going to release what's called leukocytosis inducing factor. That'll go to the bone marrow and it will stimulate the production of white blood cells. And so you'll get increased numbers of white blood cells in the bloodstream. Then those white blood cells are going to migrate to the injured area. They're going to know that they've come to the injured area because of cytokines that will be released by the injured tissue, acting as neon signs that say, here's the infection, or here's the injury, stop here. So then they're going to stop in a process called margination. Typically, macrophages, white blood cells, are going to roll along the endothelium of the blood vessels, kind of like Velcro, hooks and loops, but they're very, very temporary and they're very easily pushed along by the flow of blood, but they're going to roll on the inside of the endothelium. But when tissue injury has happened, cytokines will cause those endothelial cells to secrete what are known as ICAM proteins. And then on the white blood cells, on the macrophages, then they will stick up integrin proteins that will then link tightly with the ICAM proteins and cell adhesion molecules, and then hold those macrophages in place. And then more chemotactic factors or more cytokines come and trigger the next step, diapedesis, where the cell reconstructs its cytoskeleton so that it becomes very flat and can pass through the capillary walls, and then the macrophages and the neutrophils will migrate to the site of the injury, and phagocytosis of the pathogens and any dead tissues will happen, notice, by neutrophils in the short term and by macrophages in the long term. Pus may form, and then the macrophages will clear the area of debris, and then healing can happen. All right, but that's not inflammation. So let's go to inflammation. So what's going to happen, again, tissue injury is going to trigger mast cells, which are typically out in your tissues, out in your skin. It can trigger complement proteins. We'll get to that. Other chemicals like the kinins, bradykinin, and the other one I can't think what it's called, and then prostaglandins. And so all of those are inflammatory chemicals. And the chief inflammatory chemical of this group is histamine. So what does histamine do? Well, it causes vasodilation, and it causes increased vascular permeability. So these are the two things that histamine does. Now, why would we want to do that? We'll get to that in a minute. And also, these chemicals can do the chemotactic factor, act as chemotactic factors, and bring the leukocytes, the white blood cells, to the injured area. But these are the two things that I want to focus on right now, is the vasodilation and increased vascular permeability. You're probably familiar with the 4 hallmarks of inflammation. They are heat, redness, pain, and swelling. So 2 of these are caused by vasodilation. So what vasodilation does is cause local hyperemia, or increased blood flow to the area that is going to make the area warm, and it's going to make it red. Now notice with the heat, oh yeah, there's an effect of that, but there's nothing with redness. Redness, it just identifies to you, because of that increased hyperemia, that there's infection there. Okay, so you know, you've seen how red an infected body part gets. But redness doesn't do anything, but the heat most certainly does. And so by increasing the temperature, of course, because your temperature is regulated by your blood, this increased local temperature increases the metabolic rate of cells so that healing is promoted. So mitosis is going to happen at a much rapid rate if it's hotter, if it's warmer. And so we want to warm up the area. So why do we put ice on things? I'll get to that in a minute. Okay, then I don't know. Maybe I won't discuss that. I haven't decided. I don't know that I got time to do that, because that's a sore spot with me. Anyway, okay, so then what else do we get? So we get increased capillary permeability, our capillaries, and actually it's not our capillaries that will leak fluid, but the post-capillary venules. So just after the capillary bed is what gets leaky, and that's where the fluid that's going to come out, exudate, because it's leaving the bloodstream. And it's really plasma that's coming out. Now, in our plasma, we've got a couple of things that are going to come out here. One of them are protein-rich fluids, and so now we have nutrients and things that the cells will be able to use to heal. But along with that leakage, we're going to get swelling, obviously, and we're going to get pain due to the swelling and the stimulations. The bradykinins are often pain stimulators. So why do we want pain and inflammation? Well, it's to make us stop doing stupid things so that we don't do it again. Now, we didn't necessarily do a stupid thing, but it's to let us know, don't keep using that inflamed body part. And then swelling is because we want that nutrient-rich fluid out to the area to nourish the cells to promote healing. Now, another benefit of this fluid that comes out of the postcapillary venules, is that there will be clotting proteins. So the same clotting proteins that we saw in inflammation, then in coagulation, are the same ones that will be out here in the tissues. And what they're going to do is wall off the area and try to contain the germ, the pathogen, at the portal of entry. We do not want that pathogen going somewhere else and infecting other healthy tissues. And so walls off the area to prevent injury to the surrounding tissue. And then the temporary fibrin patch also forms a scaffolding for repair to promote healing. So that in a nutshell is how inflammation works. So it's triggered when the body tissue injury occurs. Why do we have it? Prevents the spread of the infection or microbes, disposes of debris and pathogens, and enhance the repair process. Again, redness, swelling, heat, and pain, sometimes impairment of function. So here are the chemicals that are released, and we're going to get into complement in just a minute. And then hyperemia. So when the tissues get congested with blood, we're going to get heat and redness, and then the exudate that comes from that is going to cause the swelling and pain. Now, here's another reason why we have edema, why we have swelling, is to help dilute harmful substances as well. And there's the nutrients and then the clotting proteins to isolate the injured area. Here is the phagocyte mobilization or leukocyte extravasation, however you want to say that. So we stimulated the white blood cells to be made, and then they stuck to the site of infection, margination, and diapedesis. So they rearrange their cytoskeleton, so they flatten out, and then chemotaxis will cause the neutrophils to follow the chemical trail that will get up to wherever the infection is. Okay, so then I went through all of those, all of that, and so there's that. And then I already talked about what pus is. Okay, so another second line of defense is interferon. So here's what happens with interferon. It is a chemical produced by our body cells that it interferes with viral replication. But we don't start stimulating the production of interferon until we have been infected with a virus. And so here's what happens. One cell's going to get infected with the virus, and it's like throwing itself on a grenade. And so it got infected. Here comes the virus, and it injects its nucleic acid, and then instructs, and then essentially commandeers the replicative and protein translational process of the cells, and then the cell, the body cell, the host cell, essentially becomes a virus baby factory. And then once the viruses have been made, then the cell will be lysed in order for the viruses to leave. And so if you've been infected with the body cell, then you're done. You're never going to get better from viral infection if you are a body cell. Inevitably, you will be lysed and you will die. So we want to reduce the number of cells that become infected. So once the host cell becomes infected with the virus, as the viral DNA becomes incorporated into our DNA, it sits down and triggers the production of, or turns on the genes for interferon production. So here's the messenger RNA. So we transcribe the gene for for interferon, and then the cell produces, then we translate it, and we produce interferon molecules, which we package and secrete to the interstitial fluid, to our surrounding cells. So it makes the interferon, and the host cell then is ultimately killed by the virus. But the surrounding cells will then, the interferon will bind to the surrounding tissue, which will then turn on, so that interferon will trigger the production of antiviral proteins inside of the cell. And so then as the new cells become infected with the virus, they are going to block viral replication, or they will actually eat up viral DNA, viral nucleic acid, because viruses don't always have DNA. So I don't know why we don't always produce interferon. If it works so well, how come we don't just produce it all the time so that we don't have any viral infections? Well, I got an idea. I got a theory on, I don't know. I have a hypothesis. It's not even a hypothesis. I have a conjecture on that, but don't get to share that with you. Okay, so there is that. So there's how interferon works. Interferes with viral replication in neighboring cells, stimulating them to produce other proteins, block protein synthesis at the ribosomes. Activates macrophages and mobilizes natural killer cells. Okay. Now, complement. Here is complement. So floating around in your bloodstream right now, you have a bunch of proteins that are not active. Actually, let's go ahead and look at the definition, then we'll go back to that picture. Okay, so there's about 20 plasma proteins involved in complement, and when they are activated, they will amplify all aspects of the inflammatory response, so they'll kick in inflammation, make it work a lot better, as well as killing bacteria by attacking their cell membranes and causing lysis, and contributing to opsonization for phagocytosis. So how does that work? Well, when there are 3 different ways you can trigger these complement proteins. The first one is the classical pathway, when antibodies bind to the outside of a bacterium or other extracellular pathogen. The Y part, or the stem part of an antibody molecule, has a complement binding site on it. And so then complement will bind to those antibodies, and when complement binds, we will end up making this little complex right here called a C3. Then the C3 will break apart into 2 components here, C3A and C3B, which we'll get into a minute. Now, there are other things that can trigger the formation of this C3. Now, we don't want C3 formed when we don't have an infection, because it's really super powerful as causing our inflammation to happen. So we don't want C3 if we're not infected. So there are lectin or proteins that will bind to specific sugars on a microorganism surface, and that will trigger complement, or the microorganism may have a lack of inhibitors that allows the C3 process to occur. So either way, classical pathway, lectin pathway, or the alternative pathway, but this is typically the classical pathway. All right, so what's going to happen? C3A, and then is going to also, we're going to get a trigger of C5A, and these 2 chemicals are going to act like cytokines that are going to promote more inflammation happening in your body. But this other little subunit, C3B, is going to, notice, coats the pathogen surfaces, and then because it's stuck on the outside and it makes the pathogen tasty, then opsonization will happen and we'll get more phagocytosis. Otherwise, we are going to produce what's called a membrane attack complex, which is MAC. So C3B is going to trigger the formation of C6 or C5B actually, to C6 to C7 to C8 to C9. And notice that's these little proteins right here. And so they're going to sit down on the bacterial cell membrane, and or their target cell. Typically it's bacteria. And then when they sit down on here, they've produced this membrane attack complex, and now the insides are going to leak out. We've created a pore, and now the cell is going to lyse. So notice it says membrane attack complexes form from activated complement protein components, C5b and 6 through 9, that insert into the target cell membrane, creating pores that can lyse the target cell. So that's how that happens. Okay, and then the last one, and I'm going to stop here, and I'm going to make a separate video for specific immunity. Our last non-specific defense, our second line of defense, is fever. So when the macrophages are activated out in the tissue, they're going to send out additional signals that say, hey, guess what? We need some help because it's really yucky out here in the trenches, and we need your whole body to do something. And so it will secrete, the macrophages will secrete chemicals called pyrogens, which mean fire formers. And these are the fever-causing chemicals that are going to go to the hypothalamus, because that's where body temperature is regulated from. And so if you need to increase your body temperature, send the pyrogens to the hypothalamus. The hypothalamus then will ramp up your body temperature, And why do we want that? Because heat inactivates bacterial enzymes. It also keeps iron and zinc sequestered in our liver and away from the bacteria because they need that for their cellular function. And then, like I said, if you have a warmer than normal body temperature, mitosis will happen at a more rapid rate. And so that's why we have fever. And so that's why you should not, at the first sign of a fever, take a fever reducing medication like Tylenol or acetaminophen, ibuprofen. We want fever to work for a while. I have a cardinal rule in my family, and now that my kids are grown, they'll probably do the same thing with their kids. I don't give them anything if their fever is below 102.3. I don't know why I picked 102.3, because that's just when they got crabby and felt bad and didn't function well, and I didn't want them to be miserable, and I also didn't want them to get a febrile seizure and have denaturation of body proteins and stuff like that if they got a fever of 104 or 105. So I made them suffer through it, and it's good because your body is designed to produce a fever to help fight this infection. And we're still trying to keep it from being systemic, and that's the key in all of these 5 things we just looked at. We don't want the antigens to go systemic. We don't want the pathogens to go systemic, because then we got to go hunt for them. And that's why we have a specific line of defense, or a third line of defense, or specific immunity, or adaptive defenses, which I will do in the next video.